In this procedure, enzyme mixtures for converting lignocellulosic biomass to free sugars are optimized using an automated system known as a GLBRC Enzyme Platform or GENPLAT. First, a mixture model is designed in which 4 to 18 distinct enzymes are combined in up to 96 different ratios using design of experiment software. The biomass and enzymes are then pipetted into 96 deep well plates using a liquid handling robotic system. The plates are sealed and incubated for 48 hours at 50 degrees Celsius. Following the incubation, the wells are sampled using an automated liquid handling system, and the free glucose and xylose are measured using a colorimetric enzyme-coupled assay. The design of experiment software is then used to determine which combinations of enzymes yield the maximum free glucose and xylose.
The overall goal of this procedure is to simultaneously test the efficiency of various enzyme cocktails for converting lignocellulosic biomass materials into fermentable sugars for ethanol, with the overall aim of lowering biofuel production costs. Although we use this method mainly for analyzing enzyme cocktails on alkaline pretreated corn stover, it is adaptable to any pretreatment in any feedstock, such as acid-treated poplar, switchgrass, and dried distiller's grains.
This technique can be used to discover novel enzymes that are important for biomass deconstruction and to find better examples of the known enzymes.
Although the enzymes used for GENPLAT may come from various sources, the enzymes used in this demonstration are produced by expressing P-Pix or P-Pix alpha in Pichia pastoris. Grow Pichia cells in